Ladies and gentlemen, dear Pilar, welcome. It's my pleasure to announce the inaugural of Professor Pilar Martínez. Pilar, it is now 16 years ago, if I'm informed well, since the then director of the School for Mental Health and Neuroscience, Harry Steinbusch, traveled the world with a sack full of Marie Curie fellowships, searching far and wide for the most talented young researchers like a pipe people. You followed his call, and when you started here as a postdoctoral researcher, you were still in your PhD. Even though you briefly returned to Spain to finish and defend your PhD, we still managed to successfully steal you from your hometown, Valencia. In many ways, Zuid-Limburg is the Levant of the Netherlands. I can't think of any right now, but I'm sure there are many similarities. Still, Maastricht is far from the sea. No. Having grown up on the Mediterranean seaboard, you have always looked forward to going into the mountains. Now that you live here in the Dutch Pyrenees, the sea has become a holiday destination. But back to 2002, at the same time as you, your colleague, collaborator, and later partner, Mario also came to Maastricht with the Framework Program 5 grant. Who says EU-wide structural funding for research into neuroimmunology can't be romantic? Anyways, a year turned into a career. After the Marie Curie Fellowship, you successfully applied for many other grants, such as the Aspasia for female researchers and various other prestigious grants. More recently, you became the first researcher with a grant from the Western Brain Institute in Canada. I should, of course, also mention your role in coordinating the large project that received a Zon MMA Memorable grant for your research on the influence of lipids on the onset of Alzheimer. And while, of course, the milestone we are celebrating today is testament to all of your hard work, it's also especially a recognition of the groundbreaking research you have done on the role of lipids in the brain. Your work has the potential to change not only the way we treat illness, but also how we see them, such as your review on autoimmune disease as a possible cause for psychiatric symptoms. By the way, your potential has apparently always been clear. I am reminded of an event where you were presenting our rector, Rianne Letchert, asked Nanne de Vries, wouldn't she be a good candidate for a professorial career? Add to that how Harry Steinbusch spotted you in Valencia, and it seems that our talent scouting systems works very well indeed. But we are not here to congratulate ourselves, but to congratulate you, an ambitious, energetic researcher who has achieved a lot already and who has a great future ahead of her. Ladies and gentlemen, I would like to cede the podium to Professor Pilar Martínez.
So, dear Rector Magnificus, dear colleagues, friends from Europe and the US, dear family, welcome to this traditional ceremony. I am extremely pleased to accept my chair and give here today my inaugural speech as professor in neuroinflammation of neuropsychiatric disorders. Today, I hope to convince you that this research field is very exciting and dynamic. I will start with a history of immunology, and then I will explain the concept of autoimmunity and membrane receptors, followed by my personal experience in science. As last comes the story of lipids in our research. Around the year 430 before Christ, we find the first suggestions in Greece hinting at the notion that if one survived an infectious disease, the survivor became resistant or immune to any subsequent contact to a sick person. In Western civilization, until approximately the late 18th century, it was largely accepted that disease was a punishment from God or as a result of the curse of a witch. In Eastern cultures, on the other hand, it was thought that disease is caused by an imbalance in body or so-called energy channels. After Europe suffered the Great Plagues, it was believed that diseases were the result of virulent or noxious vapors. At the beginning of the 18th century, people started to make observations regarding the origin of disease, hoping to treat or prevent illness. This, of course, was all prior to the notion that disease is caused in most cases by bacteria or viruses. Historically, Edward Jenner gets the credit for the development of the first immunization process, although previously a similar procedure had become established in China around the 1700s. The technique was called Variolation and was named after the Latin word varius, which means speckle. Variolation is the deliberate act to cause a mild infection using an unmodified pathogen. Variolation was a considerable improvement on the death rate for uncontrolled infection. The variola virus causes smallpox. The risk of death if you contract the disease was about 30%, with higher rates among babies. Often those who survived had extensive scarring of their skin, and some were left blind. At that time, it was one of the most damaging infectious diseases. Smallpox has had a dramatic impact on history. It is believed that the Spanish were able to so easily conquer the Aztecs and Incas empires because the conquistadors were carrying with them smallpox, so that the Indian population, who had no resistance at all, was almost wiped out. Immunity to smallpox was achieved by introducing the fluid that oozes out from the smallpox pustules in the nose. This technique, variolation, for the transmission of smallpox as a form of limited infection, traveled to the west from China along the traditional silk trade routes to Constantinople, where it is used spread through Europe. The royal family of England had the children inoculated against the disease in 1721. But this procedure provoked severe opposition, as physicians felt it was far too unsafe. We have to wait until 1798, when Edward Jenner found it remarkable that milkmaids were protected from smallpox if they had first been infected with cowpox. We know now that cowpox is caused by a virus that is very similar to the variola virus. Cowpox can be transmitted from cows to humans, but it leads only to mild symptoms in humans. However, Jenner's observations and experiments led him to the conclusion that inoculation with cowpox could induce immunity to smallpox. For the first time, the concept of vaccination began to take roots. In this context, it is interesting to notice that the Latin word for cow is vacca, hence vaccination. Nowadays, we still use the term vaccination for the procedure to induce immunity to many diseases. Jenner's idea first made him persona non grata, both in medicine as well as socially, as his findings were in opposition to both the church and popular beliefs. Because his method was much safer than variolation, however, the use of vaccination gradually became widely accepted, and most European countries had some form of obligatory program within 50 years of Jenner's discovery. Jenner's work is considered to be so important because of his great contribution to humanity. He is also called the father of immunology, and his work is said to have saved more lives than the work of any other individual. Nowadays, the variola virus has been completely eradicated from the entire world. Although Jenner understood that disease can be caused by transferring infected liquid into humans, he could not know what was really inside that liquid that causes the disease. If we move on history, the idea that a pathogenic organism causes a disease was not fully recognized up to a few things came together. And one of those was Anthony van Leeuwenhoek's development of microscope, and the following understanding that interesting things existed that were not visible to the naked human eye. The small organisms were the causative agent of a disease was not recognized until very much late when Louis Pasteur developed his germ theory of disease. Pasteur's original interest was fermentation of wine and beer. Important issues, to be sure, certainly for a Frenchman. But Pasteur's original interest also led him to identify the organisms that cause the fermentation process. We know now that these organisms are a type of fungus or mold called Saccharomyces cerevisiae, better known as baker's yeast. The heat kills the baker's yeast. He applied his knowledge to test also diseases, and in 1878, he studied a disease called chicken's cholera. We know now that this disease is called by a bacterium, which is called after Pasteur, Pasteurella multocida. Pasteur grew this bacterium in a type of chicken soup, and he saw that this could induce the disease in chicken. He also saw that when the bacteria was first killed, the chicken did not become ill at all when they were infected. Interestingly, he also observed that the chicken that were infected with the dead bacteria became immune or resistant to subsequent infection with the live bacteria. In other words, the chicken had become immune. With these experiments, Pasteur saw that bacteria can cause disease. Pasteur was not the only one who proposed the germ theory of disease. Robert Koch in Germany was the first to isolate the anthrax microbe or germ, and unaware of Pasteur's work, he was able to show that it caused the disease. Anthrax is a rare but very dangerous bacterial infection. Weaponized anthrax spores have been produced for biological warfare already around the Second World War. Then in 1882, Koch was able to demonstrate that the germ theory of disease applied to human diseases as well as in animals when he isolated the microbe that causes tuberculosis. Modern discoveries in immunology took place in the 20th century. Two of the co-workers of Koch were Emil von Behring and Kitasato Shibasaburo. They used vaccination with the bacteria causing tetanus and diphtheria. They made the groundbreaking discovery that toxic substances produced by these bacteria could be inactivated by the blood of vaccinated animals. Blood consists of two parts, the blood cells and the blood fluid. When blood is removed from the body in a test tube, the blood fluid separates from the cells. This fluid is called serum and can be used for experiments. This is a very useful procedure which is still used to investigate the causes of disease in animals. When they injected that serum into other animals, they found that this caused protection against the toxic substance of the bacteria. This was a very important experiment because it showed that the serum of vaccinated animals contains something or a factor that seems to protect against the disease. A researcher working together with von Behring was Paul Ehrlich, who predicted the existence of certain immune bodies, which we know now as antibodies, in the serum. Ehrlich hypothesized that these antibodies were specialized molecular structures with specific receptor sites that fit each pathogen like a lock and key. The equivalent of the key will be the antibody, and the structure of the pathogen, which we now call antigen, was the lock. The observations of these three researchers led to the concept that the body has a specific defense mechanism present in the serum in the form of antibodies. They also received for these revolutionary discoveries the Nobel Prize in Medicine in 1900 and in 1908. In fact, this now can be seen as the birth of immunology as a clinical discipline. Antibodies were identified in the blood of infected patients, and the very first clinical diagnostic tests were constructed based on antibody reactions, such as those for syphilis and tuberculosis. So immunity is good for us, right? But not all is well. At least not always. Immunity can have a problem. Instead of cure a disease, it can also be a cause of disease. Indeed, Paul Ehrlich had also realized that this powerful effect of antibodies in immunity against pathogens could potentially cause severe tissue damage if the antibody will start attacking the body itself. Instead, Ehrlich called this horror autotoxicus, which can be translated to horrible self-intoxication, which he had proposed for the first time the concept of autoimmunity, which is involved in a number of dreadful diseases. Ladies and gentlemen, so far I have mentioned some of the most important discoveries in immunology. Now the time has come to discuss with you one of the subjects of my research and master's, namely autoimmunity caused by antibodies. As I have explained before, we have an immune system which uses antibodies for its defense against pathogens such as viruses and bacteria. For the sake of time, I have not explained how the body actually makes these antibodies. When we are born, the immune system learns to tolerate everything within the body. After that, in a healthy individual, the body immediately differentiates between cells that are from your own body and therefore safe, from everything else, including pathogens, and they need to be destroyed. In people with an autoimmune disease, the immune system is defective in a way that causes it to attack cells of your own, or autoimmune diseases essentially cause your body to attack itself. The problem of autoimmunity is substantial. Recent studies indicate that nearly 10% of people have an autoimmune disease or might get one later. If we look at autoimmunity in general, we can say that approximately 1.3 million of the Dutch population, or at a European level, 59 million individuals have an autoimmune disease. In this room, there must be about 10 individuals with an autoimmune disease, or might develop one in the near future. There are more than 80 types of autoimmune diseases, some of them affecting the nervous system. Proteins which are on the surface of the membrane can receive outside stimuli and are named receptors. Membrane receptors are used for the communication between cells in the body, for example, for the communication between nerve and muscle. Often in autoimmune diseases, these receptors are the target, and I will now continue to tell you how the concept of autoimmunity against cell surface receptors as antigens have developed. Van der Lennon and Patrick Carnish published in 1971 in The Lancet hypothesis describing a new pathological process to explain lesions in certain autoimmune diseases. They suggested for the first time that a pathogen immune response can be directed against a cell membrane receptor, and that interaction of an antibody with the receptor causes a block which disrupts the normal function of that cell and related cells, leading to tissue damage. This prediction became fulfilled by the discovery of antibodies in myasthenia gravis against the acetylcholine receptor in 1973 and against aquaporin 4, a water channel in the brain, in 2005 in neuromyelitis optica, a disease that is characterized with optic neuritis. Much has been learned about disease mechanisms in myasthenia gravis in the 1970s from the laboratories of Andrew Engel, John Lindstrom, Dan Drachman, and many others. This has shown that the antibodies could inhibit acetylcholine binding to receptors in experimental animals and reduce the surface expression of acetylcholine receptors on culture cell lines by a process known as antigenic modulation, cross-linking and internalization of the receptors, followed by the degradation. And perhaps most importantly, by complement-dependent damage to the receptor-containing membrane following antibody binding. Interestingly, it was also shown by Peter Mollinar and colleagues that the reduced acetylcholine receptor function induced by the antibodies might be compensated by an increase in acetylcholine synthesis and release. So the muscle will do its best to keep a functional communication with the nerve. Later in the 90s, the cloning of anti-AChR anti-acetylcholine receptor antibodies by the laboratories of Marc de Watz and Angela Vincent helped to better understand their pathogenic mechanisms. The laboratory of Socrates Sartos has developed an array of biochemical tools to especially purify specific antibodies against acetylcholine receptor so that their contribution to the disease can be assessed. In myasthenia gravis, with antibodies against the acetylcholine receptors, the thymus exhibits structural and functional changes that in some patients can be caused by the presence of a tumor or by the development of germinal centers containing a large number of B cells. Nick Wilcox, first in London and later in Oxford, investigated the cells that were involved in the thymus in the 1990s. In the first decade of 2000s, much has been learned from the laboratories of Soria Veriachnin about the role of an anti-inflammatory environment in acetylcholine receptor thymus, which attracts B cells. Ladies and gentlemen, with this, I have to come to talk about the neuroinflammation research in mastitis and how it started. So the group started in 1976 by Marc de Watz, an internist doctor by training, who after some years in San Diego visiting John Lidstrom's lab, learned there about their new animal model for myasthenia gravis. Marc came back in 1981 to Maastricht and I started working here on myasthenia. But where did it all started for me? So I was born in Valencia in May 1972. That was a very exciting time in Spain where a new constitution was approved. We changed political gears. And after the death of Franco in 1975, many things were about to change. I was the oldest of 3 children. From Pilar and Thomas, my parents, were very active and they worked together for some years when they established a school for adults in the evening after the daily work. I remember clearly that I always enjoyed very much singing and reading. I realized only later that biology was fitting me well. I studied at the Valencia University full of great teachers and researchers. I realized that biology was so amazing that I could not be doing something else. I joined during the summer the team of Jose Maria Garcia Verdugo, who later on discovered that formation of neurons occurs in adult brain. I obtained a personal fellowship from the government from Valencia to perform my PhD at the private research institute of biomedicine from Valencia. It's called FIB, in the Department of Molecular Pathology with Juan Saus. There my first contacts with the immune system and autoimmunity started, and I learned much about molecular biology. During my PhD, I visited with a short-term fellowship the Sackler School of Biomedical Science in Boston and worked with Bridget Hoover in Lyme's disease. In 2002, during the last year of my PhD, I obtained a Marie Curie fellowship thanks to Javier Steinbus, who had a Marie Curie fellowship training site. I was its first fellow. I joined then in Maastricht the myasthenia gravis group of Marc de Watz. After my arrival to Maastricht from 2002 to 2004, I worked closely with Variolation and Maurice Stassen. They were investigating the effect of increasing the levels of rhabsine, an anchoring protein of the acetylcholine receptor, using electroporation in rats prior to antibody attack. We demonstrated that this protected against the loss of acetylcholine receptors and against further damage to the muscle. In 2003, I returned to Valencia and I defended my PhD cum laude. Already the day after my PhD defense, I came back to Maastricht to work. From 2005 to 2007, we investigated whether the increase in rhabsine levels could be beneficial once the damage is present. We saw that this increase further damaged the muscle. From our work, we concluded that the lack of receptor anchoring proteins such as rhabsine during the antibody attack is an additional pathogenic mechanism in this disease. Additionally, we investigated the anti-inflammatory characteristics of human IgG4 antibodies in an animal model of myasthenia gravis using IgG4 in monkeys. Together with Sanquin and GenMap, we reported our discovery that dynamic Fab-Am exchange happens in IgG4 antibodies, which has consequences for disease mechanisms and therapeutic strategies, since at that time already several drugs were of the IgG4 type. Recently, Inga Konechny demonstrated that anti-mask IgG4 antibodies are Fab-Am exchanged in myasthenia patients. The next step was to establish in the laboratory the state-of-the-art technology to clone and characterize pathogenic antibodies. To this aim, from 2009 to 2013, during her PhD, Kathleen Frolics immortalized mature CD22 positive B lymphocytes from peripheral blood and thymus tissue with EBV and the polyclonal B-cell activator CpG 2006. This new technology led to the identification of pathogenic antibodies in myasthenia gravis, and also we found antibodies in the CS of multiple sclerosis patients. This work was done in collaboration with the laboratory of Ferla Somas at the University of Hasselt. In parallel to this work, together with Variolation, Alejandro Gomez, and Marina Damas, during their PhD, we investigated the effect of specific drugs targeting long-lived B cells or plasma cells as an experimental treatment for myasthenia gravis, drugs known as proteasome inhibitors, which they proved to have high efficacy to reduce rapidly antibody titers in animal models of the disease. This is an important development since it could rapidly accelerate the recovery of myasthenia gravis patients by means of immunotherapy, which at that moment can take several years. Recently, the EMC Plus has become the major center for thymectomies in the Netherlands. Almost every week, patients come to Maastricht to be operated by the Da Vinci robot, which has minimal surgical implications for the patients. This work is possible thanks to a fruitful collaboration with Jos Maassen and Mark de Waets. This allowed us to collect very valuable material for analysis from patients that, in collaboration with Rosen Lepin's lab, will allow us to better understand the thymus pathophysiology in autoimmunity and in cancer. In the last 5 years, we have also begun investigating the central nervous system, where autoimmune encephalitis has been demonstrated to be associated with specific serum autoantibodies. There is the fascinating possibility that autoimmune mechanisms are playing a role in a subgroup of patients with mental illness, which might open possibilities for treatment with immunosuppressive drugs. Together with psychiatrists Jim Fanos, Bar Rutten, Teresa van Amersfoort, and neurologist Rob Rule, and inspired by the work of Joseph Dalmau's lab, we started a journey towards the brain. Caroline Hoffman and Senhua Song's PhD work focus in the identification of seropositive patients with severe mental illness. Their work clearly shows that the antibodies typically found in autoimmune encephalitis, which produce psychiatric symptoms, are very, very rare in patients with schizophrenia and depression. We are investigating the possibility of autoimmune mechanisms in patients with neuropsychiatric manifestations, with the aim to improve the diagnosis and treatment. Additionally, we make efforts to understand the role of autoimmune antibodies against specific targets, for example, the neuromuscular junction, and better understand the role of the thymus in the disease. In this research line, I foresee for the future new strategies for immunosuppression by targeting long-lived plasma cells and antigen-specific therapies for the patients. So autoimmunity is an important issue. This is underlined by this week's announcement that the Nobel Committee has awarded James Allison and Tasuku Honjo this year's Nobel Prize for Medicine for their work on immunotherapy of cancer by removing brakes on the immune system that normally prevents autoimmunity. Ladies and gentlemen, after this topic, now I will focus on neuroinflammation caused by lipids. As I mentioned before, the immune system involves many mechanisms other than antibodies, which might play an important role in the brain. So in the neuroinflammation group in Maastricht, we investigate neuroinflammation in neurodegenerative diseases in contexts other than autoimmunity. Neuroinflammation plays an important role in the majority of neurodegenerative diseases, as for example, Alzheimer's disease. But unfortunately, we do not know, or we do not understand well what is going wrong. The genetic mutations in the proteins have been studied for decades, and this has yielded some major important discoveries. Only in the last 30 years, we are starting to understand how specific lipids can affect and activate the immune system. When I ask medical students about what they know about lipids, many of them will answer that all lipids are fats. They will start thinking about fats as associated with greasy food and obesity. Fat is not popular. It's not popular in daily life, and it's not popular not even in science. But one will start wondering that if all lipids are not fats, what are they then? Lipids defines a diverse group of biological compounds, including fats, oils, hormones, and certain compounds of membranes of cells, and they are grouped together because they do not mix with water. One type of lipid, the triglycerides, are very well known. Everybody knows them. They are stored as fat in adipose cells, which serve as the energy storage deposits for the organism, and they also provide thermal insulation. The lipids I am studying form part of the membranes of cells and organelles, structures within cells, and they are called sphingolipids. Besides having a role in giving a structure, they have many other functions. So important is the organization function that membranes and lipids that form them must have been essential to the origin of life itself. They are like the walls and doors in a house, without which a house will not be a house. Sphingolipids are especially abundant in the nervous system, where neurons have to connect long distances for carrying nerve impulses from one part of the body to the other end. But where did all started with the lipids? Johann Trudikon was a German physician and biochemist. Trudikon was a pioneer of British biochemistry and a founder of brain chemistry. He's credited with conducting chemical analysis of over 1,000 human and animal brains. In his research, he isolated and characterized numerous compounds of the brain, such as cephalin, sphingomyelin, galactose, lactic acid, and sphingosine. In 1884, he explained his findings in a publication titled A Thesis on the Chemical Composition of the Brain. Only after his death, Trudikon's discovery were realized to be important scientific discoveries to the study of chemical and molecular composition of the brain. Now we know that membrane lipids are ubiquitous structural lipids in cellular membranes and potent regulators of critical biological processes. In the brain, they are abundant present in different cell types. To guarantee optimal neuronal function, the proper balance of lipids, associated metabolites, and lipid transporters are tightly regulated. As a consequence, alterations of this delicate balance in lipid metabolism contributes to the development of disease. But why this interest and why now? During my years as a PhD student in Valencia, as I was investigating Wood-Pastor syndrome, our team discovered a human protein which was interacting with the antigen of the autoimmune disease and was then given the name of Wood-Pastor antigen binding protein, and the abbreviation resulted in the tongue-twisting abbreviation, GPBP. One of my first tasks as a PhD student was to find the rat, bovine, and mouse form of GPBP. We were lucky to discover also another form of this protein, which lacks an exon of 26 amino acids. Hanada's team from Japan later discovered that this is the only protein able to transfer the lipid ceramide between membranes in cells, and therefore it was renamed in 2003 as the ceramide transporter protein in short, CERT. When I came to Maastricht, I was still puzzled that CERT was in fact a unique lipid transporter, so I continued working on it. From 2008 to 2016, the PhD students Chiara Mencarelli and Gerard Bode helped to characterize the CERT proteins in the brain. We found that CERT was present in amyloid plaques in Alzheimer's disease brains, but also it plays a role in inflammation, since it is able to bind C1q and activate the complement system, which is essential for clearance of pathogens and is one of our first arms of immunological defense. During those years, Erhard Wieberich's lab in Georgia, Augusta, saw that in animal models of Alzheimer's disease, there is an increase in ceramide levels, and this could affect the brain environment and cause brain cells to die. Also during those years, Michel Milke and collaborators from Mayo Clinic Rochester reported that an increase of certain ceramides in plasma could predict cognitive decline and the development of Alzheimer's disease. There are several questions here which are important. What is the connection of the lipids with inflammation? And how is lipid dysregulation connected to neurodegenerative diseases? Can specific lipids, for example, ceramides, be beneficial and anti-inflammatory? To answer these questions, we have undertaken in the last year's experiments to modulate certain sphingolipids and CERT in the brain of animals with Alzheimer's disease. In this way, we can assess especially which contribution they have to the non-pathological features of the disease, such as amyloid plaques, gliosis, and behavioral abnormalities. Additionally, we have analyzed the levels of these lipids over time in animals with Alzheimer's disease carrying the ApoE4 genotype, which is a risk factor for the disease. In this way, we can reveal which lipids are associated with an increased risk factor. We have also analyzed the presence of inflammation and certain lipids in rare forms of dementia to better understand the relation of the lipids with the pathology. Finally, we have investigated the effect of modulating CERT levels in the brain of animals with Alzheimer's disease. We are currently finalizing the analysis of these experiments, which I expect will help us move forward in our understanding of neurodegenerative diseases. So what we expect is to reveal a clear picture on how a disturbed lipid balance may be causative for neuropsychiatric disorders, and targeting lipids, lipid transporters, and their metabolites is of fundamental importance for the early diagnosis, prevention, or even treatment of this type of diseases. I would also like to mention the fundamental role of education in my research field. Education is one of my important duties. I have enjoyed establishing a new specialization track in pathophysiology and inflammation, which focus in understanding diseases and also in engineering the immune system. A large number of students find our specialization very appealing, and they really enjoy learning here. I also enjoy very much to educate the students in the field of lipids, since it's so much to learn there. We have an ample plan in how to improve multidisciplinary education in Maastricht, which I hope to contribute substantially in the next years. Now, I have come to the time to thank the people that have made this possible for me. So, Rector Magnificus, colleagues, students, friends, and family, highly esteemed listeners, please allow me to say a few words of thanks to the Executive Board of Maastricht University for trusting that neuroinflammation in neuropsychiatric disorders is an important research field. Professor Marc de Watz, dear Mark, thank you for your support and the opportunities you gave me since the moment I joined your group and I started work in myasthenia gravis, and also for your support in later steps of my career when I started the journey from the muscle to the brain. Professor Wim Buurman, dear Wim, thank you for a pleasant collaboration. You have been like an open book, and you have given me very important advice for my career. Professor Harry Steinbus, dear Harry, thanks for your support and vision in multicultural education and for making it possible for me to come to Maastricht in the first place. Professor Bart Rutten, thank you for your support since my first day in the Netherlands and all the things we have learned together through the years. Professor Jim van Os, Professor Teresa van Amersfoort, Dr. Machtel Marcellis, dear Jim, Teresa, and Machtel, thank you for your inspiration, support, and collaboration. Dear Rob Rule and Dr. Jan Damaso, and all the colleagues from the Autoimmunity Epilepsy Group in Maastricht, thank you for the fruitful discussions and work together. The Thymus team, a special thanks to Professor Jos Maassen, Florid Marcuse, Mirurgia Abdulhamid, Dorit Rentespies, and Professor Axel Suhausen. Colleagues from the FHML Talent Group 2017-2018, especially also for your support, for your feedback, and our joint reflection moments, and also thanks to the excellent trainers, Jim and Jos. Colleagues from the Maastricht UMC Plus Research Committee COP, especially Marco Vendes and Jacinthe Temel, thanks for the dedication and our joint activities to put science even better on the map. Colleagues from the Eurolipids Consortium, thank you for the exciting and didactic work between Hasselt, Aachen, Liège, Leuven, and Maastricht. Professor Helga de Vries, Professor Jochen Walter, Dr. Monique Mulder, Dr. Dusan Berkes, and Dr. Matthias Bouwens, thank you for our nice work together. Professor Edgar Bieberich, Professor Gergil van Echter Dekker, Professor Michel Milke, and Professor Johannes Arts, and other friends crazily interested in lipids, thank you for the nice discussions and the work we have done and still do together. Teaching colleagues, thank you for learning together along the way for your creativity and good work. Subsidies from the Brain Foundation, the European Union, the Western Brain Institute, Alzheimer Netherlands, Beatrix Fons, SONMB, and NWO, thank you for your support and vision in this research. Health Foundation Limburg, thanks for the input, feedback, and support. Special thanks to the patients and family members, thanks for the participation in the studies and scientific and practical advice. To the colleagues of the antibody platform, thanks to the efforts to establish cutting-edge antibody technology here in Maastricht. Thanks to the Myasthenia Gravis Consortium, national and international collaborators, and especially to the new generation members. Thanks to the neurologists from collaborating labs in nervous system disorders, especially thanks to Professor Joseph Dalmau, Dr. Martin Titular, and Dr. Harald Prus. Special thanks to the scientific team, especially Deborah Opteinde, Truda Driessen, Anita Finke, Via Canfors, and Anna Manet. Also thanks to all participating members and students who have helped us and continue doing so. Thanks to the Department of Legal Affairs of Maastricht University, and especially to Susanne ten Hoeve for her help and good work. Thanks to the entire Department of Psychiatry and Neuropsychology, especially the secretaries, the ICTs, thank you for your support in managing all activities. The members of the Board and Business Office of the School Mental Health and Neuroscience, thanks for all the support and collaboration, especially to Tom van de Kromenacker, Laurent Louis, Ankie Hoskenbar, Danny Langhoff, Sjur Gusters, Danny Luciana, and Marco van Herchoy for their help during the years. Division 3 of the School for Mental Health and Neuroscience, all analysts and seniors, thanks for the successful collaboration. Thanks to Rachel Caponi, who manages the Secretary of Neuroscience Division very well. Special thanks to the members of the expert group Molecular Cell Biology, Hunter Kennis, Mario Philippens, and Barbie Mahils. My previous PhD students, postdoctoral students, and the students and researchers of the Neuro-inflammation group, in particular Dr. Fernambuk, Dr. Janssen, Dr. Frolix, Dr. Gomez, Dr. Mencarelli, Dr. Bode, Dr. Stevens, Dr. Hoffman, Dr. Nogales, Dr. Saxena, and Dr. Konesny. Thanks for your enormous contribution, support, and collaboration. Thanks to the current postdoc, Dr. Caroline Hoffman, and PhD students working in our group, Simone Crivelli, Caterina Giovannoni, Kian Luo, Dan van Kreuning, Shen Huasong, and Marina Damas. Your hard work, enthusiasm, and vitality is so contagious. Thanks also to the external PhD students with whom I am collaborating with, Andreas Paulus, Sandra Den Hoed, Annalena Schathauer, Nikita van den Burg, and Nienke de Witt. Dear Peter, thank you very much for the wonderful, pleasant collaboration during the last years, during the last 10 years in the heart of our neuroinflammation group. Together as a team, we can reach very far. Special thanks to Mario. Thank you for the day-to-day interaction, for the successful work done on our current work. Your creativity and critical mind is essential to our research and the way we think in science. Friends, family, and family-in-law, especially Akim, Pia, Jan, and Nikki, thanks for your support and love. Mis padres, Tomás y Pilar, mi hermano Tomás y hermana Cristina, muchas gracias por vuestro amor y apoyo. Gracias también a vuestras familias por darnos siempre cariño y tiempo. My partner Mario and our children Sara, Sofia, and Isabel, and my partner son Sean, many thanks for your love and for our nice life together. To finalize, I will borrow the words of Nikola Tesla. The scientific man does not aim at an immediate result. He does not expect that his advanced ideas will be readily taken up. His work is like that of the planter for the future. His duty is to lay the foundation for those who are to come and point the way. I have said.
Professor Pilar, thank you for the good lecture you gave us. I think it's clear that you have a very good network here. You had so many thanks to so many people, that's absolutely a sign of it. This official ceremony is coming to an end now, and so we go to the reception. As one of my predecessors often say, Horeca est. So if you follow us, then everything will be okay. Thank you very much.